Imprinted genes is the gene for the insulin-like growth factor. Insulin-like growth factor 2. This gene encodes a growth factor that binds to its cognate receptor to activate cellular proliferation and growth of the tissues in an animal. Okay, this is a normal gene. And it was found that on mouse chromosome 7, the insulin-like growth factor gene was only expressed from the paternally derived allele. The maternally derived allele was not expressed at all. So this is one of the examples that was shown as a result of this early experiment that the genomes aren't equal. And one of the genes that is not expressed equally is the insulin-like growth factor 2. And in this case, it's only expressed from the paternally derived allele, not from the maternally derived allele. Okay? And what's interesting is the receptor. The receptor for the insulin-like growth factor that is imprinted, the gene that encodes the receptor for this growth factor, that gene is also imprinted. It's on mouse chromosome 17. And in this case, it's the maternally derived allele that encodes the insulin-like growth factor receptor. This is the one that's active. The paternally derived allele is not. Okay? And so these are what you would call imprinted genes. So what happens if you play around with these genes? What happens is the insulin-like growth factor receptor, what it does is it binds and degrades excess insulin-like growth factor 2 that's out in the bloodstream of the developing animal. And so if you delete or if you mutate and get rid of the paternally derived allele, because it's not expressed, it's not going to have the effect, right? If it's not expressed anyway, if you mutate that, it's not going to have an effect. However, if you mutate and inactivate the maternally derived allele, that's another story, because this is the allele that's actually expressed. What you get in this case is fetal overgrowth and death. Okay? Does anybody have a question? All right, we're going to look at some pictures now. All right. Just looking at the insulin-like growth factor 2 gene. We're not going to look at the receptor that I described here. But we're just going to look at this gene right here. Actually, in mice, the insulin-like growth factor gene is expressed from the paternal chromosome number 7. It's not expressed from the maternally derived chromosome number 7. And so you only need half the amount of protein. Therefore, one of these is not one of the alleles, the maternal allele is not expressed, and you get normal mouse development. It's called wild type, remember? Wild type means normal. If you mutate the female derived allele, but because it's not expressed, it's not going to affect the mouse at all, right? If you had a test question like this, would you be able to follow it? All right. If it's not expressed and you mutate it, what's going to happen? Nothing. Absolutely nothing. However, if you mutate the allele that is expressed, you're going to have an effect, right? And in this case, they mutated the allele that's expressed, of course, from the paternally derived chromosome 7. So you didn't have proper levels of insulin-like growth factor in the developing animal, and you got a dwarf mouse. Okay? I'm sure he looked really cute. All right. We can deal with a phenotype like that, right? That's okay. All right. In humans, we actually have diseases associated with imprinting And a number of these diseases that are very well known are Beckwith-Lehman syndrome, Prader-Willi syndrome, and Angelman syndrome. I'm going to talk about Prader-Willi syndrome and Angelman syndrome. The genes that are involved in Prader-Willi syndrome and Angelman syndrome are on human chromosome 15. They're on human chromosome 15. Most of us have you have to have normal human chromosomes 15, right? A lot of important genes are encoded on chromosome 15. And so most of us will, when we go to have children, you have a normal chromosome 15 in the mom, a normal chromosome 15 in the dad. You get a normal wild-type individual walking around. And we are wild, right? Yes? Okay. All right. If, however, there's a region in chromosome 15, if that's deleted or mutated, what's going to happen is if this is deleted or mutated in the female-derived chromosome 15, and this happens during oogenesis, or formation of the egg, what's going to happen is when she goes to mate with a male, when that woman who's carrying a blighted egg with a deleted set of chromosome 15 in her egg, when she mates with a normal male, their child is going to have Angelman syndrome. Angelman syndrome is associated with profound mental retardation. These children never develop properly. They are nonverbal. These are the kind of individuals that when you go to the mall, you feel very sad because these are the children that are in these big wheelchairs, being pushed around. They're nonverbal. They're profoundly retarded. They can't walk. They can't talk. They have what you would call minimal ability to interact. That's Angelman syndrome. Thank heavens it's relatively rare. If, however, if that's Angelman syndrome happens when a woman during oogenesis sustains a mutation in this segment of chromosome 15. And if she ovulates the egg with this mutation in chromosome 15 and mates with a normal male, the child will have Angelman syndrome. What's interesting is if in the male, if the abnormality on chromosome 15 happens in the male during spermatogenesis, and the abnormal sperm carrying the abnormality on chromosome 15 mates with a normal woman, the child they'll have in this case has Prader-Willi syndrome. Prader-Willi syndrome children are mildly retarded, but there's a problem in their ability to control their appetite. There is no such thing as satiation. They will literally eat themselves almost to death. Not their fingers and their lips, that's Leishmanian. But I'm just talking about families where you have a Prader-Willi syndrome child. They literally have to have padlocks on the freezers, the refrigerators, the kitchen cupboards. These children cannot get enough food. There's a problem with appetite control. That in itself affects every aspect of their lives. Okay? I know. There are people in here who think, well, I have that issue. Come lunchtime, I cannot be satisfied. But no, believe me, you do not have, if you're walking around, you do not have Prader-Willi syndrome. Okay? All right. Prader-Willi syndrome is a severe syndrome. Again, they're mildly retarded, but there is just no such thing as enough food for these people, and it drives every aspect of their lives, and it changes the life of their families extensively. Okay? So what on earth, and what I can tell you also, is that if you have an egg that has the deletion in chromosome 15 and a sperm with the deletion in chromosome 15, that ends up as an embryonic lethal. Okay? So everybody in this room, when it comes to chromosome 15, we're all wild type. Okay? We're all wild type, because if you were carrying this mutation, you would know it. There's nothing subtle about this. So what on earth is it that's in this region of chromosome 15 that has such a profound effect on development and behavior? One thing involves, in the case of Angelman syndrome, the region that's actually deleted is a small segment of DNA. That's actually a regulatory region. It controls expression. Of UBE3A gene. The UBE3A gene is what is controlled by this little segment of DNA right here. It's a regulatory control region. It's not a promoter, but it functions as an enhancer. To activate expression of the UBE3A gene. UBE3A is a ubiquitin protein ligase. So what happens in this case is ubiquitin protein ligase in our cells, especially the cells in the brain. This particularly affects the brain. And in the cells of our brain, ubiquitin protein ligase, what this does is old proteins that are in the brain that need to be degraded. Remember, I talked about this. At the level of controlling gene expression, one of the things that has to happen is when proteins get old, they have to be degraded. And for a protein to be degraded in the brain cells, or in all of our cells for that matter, a ubiquitin protein ligase will actually attach a ubiquitin chain to an old protein. That old protein will then be degraded in the proteasome that's present in our cells. So in our cells, when proteins get old and the cell needs to dispose of it, a ubiquitin chain is attached to the old protein, a ubiquitin protein ligase, and then that it's called a ubiquitinated protein, is then degraded in a complex in our nerve cells and in the rest of our cells of our body. It's called a proteasome. We'll actually break it apart. So, can I repeat anything? Are we okay? Okay. So, if in Angelman syndrome, the enhancer region that activates expression of the gene that encodes this protein, this ubiquitin protein ligase, if this isn't working, then the reason why these children are so profoundly retarded and affected in their lives is because the old proteins that are present in their cells, especially the cells in their neurons or their nerve cells in their brains, there's a buildup of proteins that need to be degraded and they're not. And so that is what affects these children. That's the biochemical basis for the mental retardation that you see in children with Angelman syndrome. Can I repeat anything? Question?
Yeah, so since we're all outside, how do we get the mutated?
I'm sorry, what?
Since we're all outside, how do we get the mutated?
Okay, again, nobody with Angelman syndrome or Prader-Willi syndrome is out there getting dates and producing children, right? No, absolutely not. So the mutation occurs. The mutation occurs during development of eggs, oogenesis, or development of sperm, or spermatogenesis. That's when the mutation occurs. Because like I said, nobody with Angelman syndrome or Prader-Willi syndrome is out there getting dates and producing children. Not happening. Okay? All right. When it comes to Prader-Willi syndrome, the less what you call severe of the two imprinting syndromes from chromosome 15, Prader-Willi syndrome is an enhancer region. It's a region that's pretty close to the Angelman syndrome enhancer. Prader-Willi syndrome enhancer activates expression of a whole series of genes on, on the male-derived chromosome 15, activates a whole series of genes, the ACE2, NZN, ZNF217, small nuclear RNAs. All of these are actually under control of the Prader-Willi syndrome enhancer. So if this is not functional, these genes are not being properly expressed, and you're going to develop Prader-Willi syndrome. What's interesting is this ubiquitin protein ligase. This is only imprinted in cells of the brain. Only imprinted in cells of the brain. So the Angelman syndrome enhancer that activates expression of the ubiquitin protein ligase gene from the maternally derived chromosome 15, this only is happening in cells of the brain. That's why you have, like I said, profound mental retardation as a result. Okay. Oh, all right. Oh my goodness, let's go to something more interesting. Okay. That's not interesting, but it's just downright depressing. So let's go into something that's a little bit more lighthearted, but at the same time educational. And we all know that out in the world we have all kinds of breeds of dogs. We have Great Danes and we have Chihuahuas, right? These are the same species. Which means from 1111, if something is of the same species, can they mate and produce viable offspring? Don't want to think about that, for crying out loud. But nonetheless, conceivably, these guys can mate and produce viable offspring. Not going to be so easy. Most dog breeds that we know of have only really been developed over the last couple hundred years. It's all artificial selection, okay? Not only do dogs exhibit all kinds of size differences, rabbits do as well. This is Big Ralph. Look at the size of him. Have you ever seen a rabbit this large? He's called a continental giant. Isn't he nice? Oh my gosh. He's actually a house pet, believe it or not. Big Ralph. You can teach him to use a litter box. Well, you should have one. Anyway. They use a litter box. They don't.
We fill it up. Yeah, you
Nonetheless. Okay, so this gives you an idea. Not only dogs, but also other animals as well will exhibit different sizes, different phenotypic attributes. So let's take a look at what might contribute to the size difference in all these wonderful animals that we can appreciate. So the domestic dog, as we know it, actually descended from the gray wolf only about 15,000 years ago. And it's known that in the 5,000 years from the time that the domestic dog was descended from the gray wolf, even to graves in the Middle East and Europe, about 10 to 12,000 years ago, by that time, small terrier-sized dogs were appearing. And these terrier-sized dogs just came on the scene as a result of mutation. In the genes that are associated with size, and it was through artificial selection. Who doesn't like a little lap dog, right? Okay, so it was artificial selection that resulted in the appearance of terrier-sized dogs not long after the first domestic dogs came from the gray wolf. And this happened in China. This actually happened in China. And so, based on these observations, a group of geneticists started to look at what are the genes associated with such a vast difference in appearance in an animal. And one of the genes they started to look at was, of course, the insulin-like growth factor gene. Okay, we just looked at insulin-like growth factor in mice, so there's a lot known about this pathway. So they started looking at the genes. And this gene is on dog chromosome number 15. And what they found, I've always told you about how for every gene we have 2 alleles, one from our mother, one from our father. But across the animal kingdom, you're going to have what is called allelic variation. Allelic variation. And what they found, looking at the insulin-like growth factor 1 gene, looking at the DNA sequence of the exons, the introns, the regions flanking the gene, what they started to do was a statistical analysis of changes in the gene encoding the insulin-like growth factor, and they found that there are at least 12 alleles for the insulin-like growth factor gene. What distinguishes one allele from the other is just changes in the DNA sequence. And from looking at the DNA sequence from all of these different small and large dogs, they found there are at least 12 alleles